Welcome to General Biology. Today's presentation is going to be on proteins. Proteins are made up of carbon, hydrogen, oxygen, nitrogen, and sulfur. So we added a couple of molecules compared to sugars and lipids. They're going to make up almost every structure in our body. They're going to be involved in protecting us against disease with antibodies. And they make up enzymes, which are going to be little molecules that are going to speed up reactions in our bodies, which are super important. These are some protein-rich foods. So meats are rich in proteins. A lot of seeds and nuts are rich in proteins. Eggs, milk, any milk products like cheese, and cottage cheese, and yogurt, and those types of things are all going to be rich in proteins. The monomer for a protein is an amino acid. You may have heard of people saying amino acids need to be in your body so you can make proteins. But this is an amino acid. The reason it's called an amino acid is it has an NH2 group on one end, which is an amine, and it has a COOH group on the other end, which is carboxylic acid. So it kind of makes sense. Amino acid. When we are going to form a protein, we're going to have to bond amino acids together. In order to do that, we are going to lose water. So this is a dehydration reaction. We're going to bind the carbon and nitrogen together, and when we do that, that's called a peptide bond. Okay, so the bond between two amino acids is a peptide bond. When we have a bunch of peptide bonds bonding a bunch of amino acids together, we call that a polypeptide. Poly means many, peptide, so we have many peptide bonds holding all these amino acids together. The order of those amino acids in that polypeptide is the primary structure of the protein. How these amino acids interact will often cause some folding. Sometimes they will fold into what we call an alpha helix. Sometimes they will fold into what we call beta sheets. And sometimes it's just a linear structure, strand. And so this secondary structure, or how the amino acids fold, is called our secondary structure. The tertiary structure is the three-dimensional structure of the protein. So here's some examples of three-dimensional structure. This is insulin. You guys have probably heard of that. It helps us bring sugar into our cells. This is trypsin. This is involved in helping break down proteins in the small intestines. ATP synthase is involved in both cell respiration and photosynthesis. So there's just a bunch of examples. Hemoglobin is involved in carrying oxygen around our bloodstream. So these are a bunch of examples of three-dimensional structures of proteins. What jobs do proteins do? Well, they help in digestion. They help in copying our DNA into RNA, and they help in the process of making proteins. They help carry molecules around inside of our body and our bloodstream. They're involved in hormones. So hormone communication is important for proteins. Structure, every structure in your body is pretty much made up of proteins. Antibodies are made up of proteins, so we're going to help fight disease. And all of our movement happens from protein-protein interaction. You have fibers in your muscles called actin and myosin that interact with each other to cause our muscles to contract. So movement is also protein-based. One of the types of proteins we mentioned was an enzyme. So an enzyme is going to be a molecule that helps speed up reactions. And what it's going to do is it's going to bind to a particular molecule in our body. So it's going to have a site on it where a substrate will bind, and then a reaction will take place. And it will either be a breaking apart, degradation reaction, or an anabolic reaction where it builds something. So in this little example here, we're breaking sugar, okay, into two So this is a sucrose molecule. Okay, it's coming in, it's being broken, and then the glucose and the fructose, which if you combine them make sucrose, are released. Enzymes are reusable, so as soon as it gets done breaking these apart, it can be used again in another reaction. Enzymes have optimal temperatures. That means there's a best temperature and there's a best pH for every enzyme. Okay, all the human enzymes work best about 37 degrees Celsius. Okay, but not all animals have the same temperature as we do. The enzymes in our bodies don't all work best under the same pH conditions, though. Our intestines have a pH of about 8. Our blood is pretty neutral, around 7. Our stomach's a little under 2. And so the enzymes that work in these different areas work best under those particular conditions. And lastly, antibodies. Antibodies are going to be involved in helping fight against disease. They have little regions on them that identify certain structures, and they can bind to those structures. And when they bind on the outside of an invader, they stop the invader from being able to penetrate our cells, and they also make it easier for our immune system to clean the invader out of the bloodstream. This is a picture of a bunch of antibodies bound to the outside of COVID. So one of the treatments we're looking at right now for COVID is monoclonal antibodies from people who have already had the disease. So we can take their blood, we can remove antibodies from it, we can cause those antibodies to reproduce themselves and get lots and lots of those from those B cells. And if we can get enough of them, then we can inject those into somebody who has recently come down with COVID, and it will help them clear COVID from their bodies. This might be an important treatment prior to the production of a vaccine. Vaccines cause a similar response inside our bodies.